In this video, we're going to be talking about glycolysis. So I'll introduce you to the idea of glycolysis, and then we're going to focus on just the first phase, the preparatory phase of glycolysis. In a later video, we'll talk about the second phase, the payout or the payoff phase of glycolysis. So what you'll need to know about glycolysis for the exam is I want you to know the names of each of the intermediates, and I want you to know the structure of each intermediate. So ideally, I want you to be able to sit down on a piece of paper without having any other materials in front of you, draw the structures of all the intermediates, name them all, name all of the enzymes that catalyze each of the 10 steps, track the carbons, and I'll talk about what that means in a moment, know which of the 3 steps are irreversible, okay, have such a negative delta G that they are under physiological conditions irreversible, and then the total balance sheet, what goes in, what comes out. So if we take a look at metabolism in general, so here we have the mitochondria, out here we have the cytoplasm, right here we have the endoplasmic reticulum, right? We've got glycolysis going on, we have the citric acid cycle going on, we have the breakdown of fatty acids. So there's a lot going on. In fact, we're going to learn pretty much this entire figure, all the reactions of this entire figure by the end of the 8 weeks of the course. But right now, what we're focusing on is glycolysis, turning glucose into 2 molecules of pyruvate in the cytoplasm of cells. Now, glucose can be used to do all sorts of things. We are focusing right here in this lecture, the oxidation via glycolysis to pyruvate. We can also use glucose to make ribose 5-phosphate. We'll then go on to make nucleic acids and also generate an electron carrier called NADPH. If we have an excess of glucose, our cells can use it to build glycogen if we're in an animal cell, or starch or sucrose in a plant cell. If we're a plant cell, we can also use glucose to make the polysaccharides of the cell wall, like cellulose. So there's lots of things that cells can use glucose to do. Right now, we're focusing on the oxidation of glucose to 2 molecules of pyruvate. Now, glycolysis is one of the most primitive forms of metabolism, one of the first forms of metabolism that evolved. Glycus means sweet, lysis, splitting. So splitting of the sweet stuff is what glycolysis means. It's the fate of most carbons within cells. Like I said, it occurs within the cytoplasm, so it doesn't require any specific organelle to happen. It's a stepwise degradation of glucose. It produces energy via the ATP that's produced and the NADH that is collected. And for organisms that live anaerobically, meaning without oxygen, and then even for some of the cells within a multicellular organism, that's the sole source of energy. Those 2 ATP that we get from glycolysis is the sole source of energy for many of these organisms and for many cells within our body, erythrocytes, renal medulla, brain, sperm cells. Now, 3 of the 10 enzyme catalyzed reactions within glycolysis are irreversible. And you can see they all have something in common. 1, 3, and 10. They all have very negative delta Gs. And what that means is technically they're reversible, but under cellular conditions, they are not reversible. That delta G is so negative, there's really no strategy a cell can use to get the reaction to go in the opposite direction. And so I have 2 questions I'd like you to ponder. What does this imply about all of the other reactions? Okay, so the other 7 reactions. And what's the significance of this? Okay, because what we're going to be looking at a little bit later is something called gluconeogenesis. And gluconeogenesis is a way to convert pyruvate, 2 pyruvates, into glucose. So how many of those enzymes can we use in just the other direction? And what do we do about the enzymes that we can't use? I'm not going to go into the answers to that, but I want you to think carefully about it. All right, we're going to focus on the preparatory phase. That's the first 5 steps of glycolysis. And we call it the preparatory phases because even though glycolysis is going to net us to ATP, in this first phase, look at this. In reaction number 1, we're spending an ATP. In reaction number 3, we're spending an ATP. Okay, so that's why we're investing. Okay, hopefully in those next 5 reactions, we're going to get a payoff, and we will. That's why it's called the payoff reactions. But in this step here, we are preparing. What we're doing is we're taking this glucose molecule, we're slapping 2 phosphates onto it, okay, and then we're splitting it into 2 molecules of glyceraldehyde 3-phosphate. Here's molecule number 1 of glyceraldehyde 3-phosphate. Here's glyceraldehyde. Here's molecule number 2 of glyceraldehyde 3-phosphate. Now, when you track your carbons, when I mention tracking carbons, what I meant is this. We can number each one of the carbons in our glucose molecule, and they're numbered in a very specific way. Here's carbon number 1, 2, 3, 4, 5, and 6. And as we go through the reactions, I want you to be able to keep track. Which one is 1? Which molecule is 2? Which carbon is 2? Which carbon is 3? And what you'll notice here is that we have a 6-carbon molecule and we break it down into 2 3-carbon molecules. Right? So glyceraldehyde 3-phosphate is a 3-carbon molecule. And we get 2 of those from 1 6-carbon glucose. All right, let's number those carbons. Well, this first glyceraldehyde 3-phosphate is from carbons 4, 5, and 6. Over here, 4, 5, and 6. Our other glyceraldehyde 3-phosphate is from glucose carbons 1, 2, and 3. That's from these carbons here, 1, 2, and 3. Okay, so this is what I mean by tracking your carbons. You're always going to figure out, okay, where is carbon number 1 from glucose? Where is that now? Where is that now? Where is that now? And I want you to be able to do that for all of the intermediates. So let's start talking more specifically about these 5 reactions. The first one is turning glucose into glucose 6-phosphate. Right? This is very straightforward. Right? If you can draw glucose and then say, all right, we're turning it into glucose 6-phosphate, you should easily be able to draw glucose 6-phosphate. You should be able to derive the structure. It's just basically taking this oxygen here and now connecting it to a phosphate group. Of course, that is going to require a molecule of ATP. And remember, when we're dealing with reactions that transfer a phosphate group to or from ATP onto something else, we use a kinase. In this case, we're using hexokinase because glucose is a hexose. And here we have to track our carbons. So carbon 1, 2, 3, 4, 5, 6. Now, this reaction, look at this, under standard conditions, right? If we're looking in specific cells, our delta G is negative 33.4. This is an incredibly exergonic reaction, so it's one of our examples of an irreversible reaction. And again, the consequences of this are twofold. Number 1, it means that under cellular conditions, we cannot use hexokinase to turn glucose 6-phosphate into glucose. And number 2, the other consequence that we will get into in a lot more detail is that because this is an irreversible reaction under cellular conditions, hexokinase is a regulated enzyme. It is an enzyme that is allosteric that will allow us to turn it on and turn it off within the cell. Now, there's one more thing. This brings up a great example. Why are intermediates phosphorylated? Well, the first reason is that as soon as glucose is phosphorylated to glucose 6-phosphate, bang, it's trapped within the cell. Can't get out of the cell. It's there's no carriers. It certainly can't get through the membrane of the cell, but then there's no carriers that allow glucose 6-phosphate through. There are carriers for glucose, but once glucose gets in, it gets turned into glucose 6-phosphate, can't get out anymore. These high energy intermediates then can, these phosphorylated intermediates can eventually transfer their phosphate to a molecule of ADP to make ATP, and we'll see that a little bit later. And then there are enzymes that can only work, that have active sites that only interact with the phosphate groups of these phosphorylated molecules. Okay, reaction number 2. Reaction number 2, all we're doing is turning glucose 6-phosphate into fructose 6-phosphate. We haven't taken away any molecules or atoms, we haven't added any atoms, we're just we are isomerizing this. And so therefore we use an enzyme called an isomerase. And because we are isomerizing glucose 6-phosphate, it is called glucose 6-phosphate isomerase. So it's very, very straightforward. It's very easy to remember, okay? So here what we have is we've got our glucose 6-phosphate, and if we change it to the chain form, it looks like this, and we have an aldose here on that one carbon. All we're doing is changing that aldose to a ketose. So we are moving that carbonyl to carbon 2, and there it is. Okay, so it's identical. So now we have to put it back into the ring form, and so we've got to attack that carbonyl carbon, and which hydroxyl group do we use? Right there, carbon number 5 hydroxyl group. This O attacks that C, and what that means, it's going to be a 5-membered ring with 2 carbons sticking out of it. Carbon number 1 and carbon number 6 are going to be sticking out of that ring, and there it is. So here's our 5-membered ring, 1, 2, 3, 4, 5. And there's carbon number 1 sticking out and carbon number 6 sticking out. And that carbon number 6 is what's connected to the phosphate group. Thus, we have fructose 6-phosphate. Now notice, look at this, under cellular conditions, it has a delta G of negative 2.5, very close to 0, which means all it takes is just a little bit of a boost to the products or reduction in the reactants, and suddenly you've got that reaction running in the opposite direction. Okay, reaction number 3, and this one is catalyzed by a very important enzyme called phosphofructokinase 1, PFK1. It is another kinase, which means here's our second ATP that we are spending, and we're spending it on phosphorylating the 1 carbon. So what we end up with is a molecule called fructose 1,6-bisphosphate, which is super easy, right? We've already drawn fructose 6-phosphate, and now we're adding a phosphate group to 1, and so it should be very, very easy for you to derive the structure of this molecule, okay? Now, again, it's an irreversible reaction. It's our second of 3 irreversible reactions at negative 22.2 kilojoules per mole. Now, you guys don't have to know the exact numbers of the delta Gs, but you should know which ones are very negative, right? Those 3 that we discussed. Okay, so now we've got this molecule, fructose 1,6-bisphosphate, right? Very high energy molecule. The next step is our lysis reaction. When we talk about glycolysis in the word glycolysis, well, this is that lysis reaction, and it's catalyzed by an enzyme called aldolase. And what that's going to do is it is going to break the covalent bond between carbons 3 and 4. And so what we've done here between this slide and the previous slide is we've opened up that fructose ring back into its chain form. So notice there is our carbonyl carbon, our ketose of our ketose. There's the 1 carbon with the phosphate group, there's the 6 carbon with the phosphate group, and all the carbons in between. And now what aldolase does is it catalyzes the breaking between 3 and 4. And so what we end up with are 2 3-carbon molecules, 2 different 3-carbon molecules. Dihydroxyaldones. acetone phosphate. Now we can use that name to deduce or derive what the structure is going to be. We know it has 3 carbons, so we draw 1 carbon, 2 carbon, 3 carbons. We know carbon number 1 right here is phosphorylated, so that's easy as well. And now let's read the name, dihydroxy. Okay, dihydroxy, meaning we have a hydroxyl group here. We've got a hydroxyl group that phosphate group was attached to, and then acetone, dihydroxytone acetone. Acetone is a ketone, and there's only 1 carbon to make it a ketone, this middle carbon. If that carbonyl was at the 1 or the 3, it wouldn't be a ketone, it'd be an aldehyde. All right, so you can use the name of these intermediates to help derive the structure. Same thing here, glyceraldehyde 3-phosphate. Okay, well, we know we've got a phosphate group here on this 6 carbon. Glyceraldehyde, it's got to be an aldehyde, so that carbonyl's got to be here on this 4 carbon. Now this 4, 5, and 6, right, that derives from carbons 4, 5, and 6 from the glucose molecule. And then glyceraldehyde, glycerol. Remember, glycerol is a 3-carbon molecule with a hydroxyl group bound to each one of them. That's from the backbone of making a fat. Well, that means that last carbon that we haven't dealt with has got to have a hydroxyl group attached to it. So using the names of the intermediates will allow you to derive their structures if you just use a little bit of knowledge from what you've learned in organic chemistry and learned about various organic molecules in past courses. And this could help you when you draw out all 10 structures. If you memorize their names, you can kind of use those names to derive the structure. Okay, but now we got a problem. We have 2 molecules that are different, and so that means if we didn't change anything, we have to have 2 different pathways to deal with them, and that is not very efficient. So what has evolved is just one more reaction here for our preparatory phase. Reaction number 5, all that does is it converts this dihydroxyacetone into another glyceraldehyde 3-phosphate. So let's take a look at that reaction. It's reaction number 5. It is an isomerase, because that's all we're doing here, is we are isomerizing dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So it's called triose phosphate isomerase. And here is from glucose, here's our carbon 3, our carbon 2, our carbon 1 of dihydroxyacetone phosphate. This molecule right there. And when we do that, we turn it into another glyceraldehyde 3-phosphate. So now we have 2 molecules of glyceraldehyde 3-phosphate. One of them that derives from carbons 4, 5, and 6 of glucose, and the other one that derives from carbons 3, 2, and 1 of glucose. So by the end of the preparatory phase, what we have done in 5 reactions and 5 enzymes is we have taken glucose, 1 molecule of glucose, and 2 molecules of ATP, and we've converted them into 2 molecules of glyceraldehyde 3-phosphate, and then our waste products, if you will, of 2 ADP that will get recycled eventually back into ATP in a different form of metabolism. So again, here's what we've done. Here is our fructose 1,6-bisphosphate. We have turned it into dihydroxyacetone phosphate and our first molecule of glyceraldehyde 3-phosphate. And then with that isomerase, we've turned this dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate. So there is our 2 molecules of glyceraldehyde 3-phosphate. So now in reactions 6 through 10, each one of those reactions that we look at is going to occur twice. Once for this glyceraldehyde 3-phosphate, once for this glyceraldehyde 3-phosphate.